Well, good morning, everyone, and welcome to this week's installment of the Fralin Biomedical Research Institute's Pioneers in Biomedical Research Seminar. It's my great pleasure today to be able to introduce our guest and speaker, Dr. Lihong Wang from Caltech, where he's the Bren Professor in the Department of Medical Engineering and Electrical Engineering. Dr. Wang did his undergraduate work and master's in optics engineering at Wang University of Science and Technology. And then he did his doctoral training at Rice University, working in the lab of Robert Curl, Richard Smalley, and Frank Patel. And that was just before the time that both Curl and Smalley won the Nobel Prize for their seminal work on buckyballs, of course. Dr. Wang went on to assume faculty positions initially at the University of Texas MD Anderson Cancer Center, and then on to Texas A&M, where he was assistant associate and then full professor, then became an endowed professor at Texas A&M. He then spent time at WashU, where he was the Beard Distinguished Professor of Biomedical Engineering and Radiology before coming to Caltech several years ago to become the Bren Professor there. Dr. Wang has made numerous important contributions using engineering technology for imaging that can be applied in all kinds of systems and areas of research and medicine, from cancer research to brain research. He serves as the editor-in-chief of the Journal of Biomedical Optics. He's an elected member of the National Academy of Engineering and a fellow of the National Academy of Inventors. He's a recipient of the NIH Director's Transformative Research Award and the NIH National Cancer Institute Outstanding Investigator Award. He also is recognized by the Optical Society of America for their award for his invention of the world's fastest two-dimensional received camera for enabling real-time imaging of fast phenomena like light propagation. I noticed that his work's been cited close to 100,000 times, appropriately so. Not only has numerous publications, over 500, but he's written a book called Biomedical Optics, Principles in Imaging. Dr. Wang's research focus is on a variety of innovative technologies for biomedical imaging. For example, he developed photoacoustic imaging, allowing looking in non-invasively into biological tissue, increasing the penetration of several orders of magnitude, so really breaking through the optical diffusion limit. He's developed photoacoustic tomography, three-dimensional photoacoustic microscopy, optical resolution photoacoustic microscopy, and photoacoustic reporter gene imaging, for example, amongst many other things. He's invented this compressed ultrafast photography of the world's fastest camera, and I think the frame rate is like trillions of frames per second. He can correct me, but it's big, fast. And photoacoustic imaging is an important modality, and many of us here in biomedical research are using technologies related to this, and I think we have a lot to learn today from Dr. Wang. So without further ado, it's my great pleasure. Please join me to welcome Dr. Wang to our seminar program. Welcome.
Thank you so much, Dr. Friedlander, for the very kind introduction. Today, I'll be talking about two different areas in my lab. One is on depth. We try to improve imaging depth in biological tissue. The other is speed. We call the second technology compressed ultrafast photography. Now, first of all, why optics? Optics is very special because of the molecular specificity. Light is very sensitive to molecules. If you look at the entire EM spectrum, electromagnetic spectrum, light occupies this tiny region, that's the only region where EM wave matter interaction takes place at the molecular level. Given the fundamental role of molecules, we really have to work on this technology. By detecting molecules, we can provide in vivo functional imaging analogous to functional MRI, in vivo metabolic imaging similar to PET, in vivo molecular imaging of gene expressions or disease markers, even in vivo label-free histologic imaging without tissue excision. But we face daunting challenges because the world is turbid in general. All materials are scattering or turbid to some extent. Even water and air we breathe are turbid. It's just a matter of extent. If you look at biological tissues, they're even more turbid. With the exception of our eye, most tissues are extremely turbid. This movie demonstrates how turbid, how scattering biological tissues are. I'm only plotting a couple of millimeters into biological tissue. If you launch a pulse, no matter how collimated your light beam is, like a laser beam, you'll see how quickly light diffuses. At the very shallow depth, you'll see that the photons travel together as a group. But within a millimeter, photon propagation transitions into the so-called diffusive zone, diffusive regime. Then it's very similar to heat diffusion. If you look at the history of optical imaging, starting more than 350 years ago, conventional microscopy was invented. It gives us really fantastic resolution, spatial resolution. We can see organelles, subcellular structures, but the penetration is limited to less than 100 microns. This is something called aberration limit. We stagnated at this limit for hundreds of years until modern light sources were invented, such as laser. Using the coherence properties of modern light sources, we're able to break through this limit, the aberration limit, and advance the penetration by nearly order of magnitude. Now we're talking about 1 millimeter penetration. Again, we couldn't advance further from this limit, something we call the optical diffusion limit, until photoacoustics was invented to provide much deeper penetration and still provide high spatial resolution. We've advanced the penetration by nearly 2 orders of magnitude. We're now talking about centimeter scale penetration for human organ level imaging or whole body small animal imaging. We do face the next challenge, something we call optical dissipation limit. Which technology can break through the next limit is still unknown. Today, I'll focus on photoacoustic tomography. Photoacoustics as a physical phenomenon was discovered by Alexander Graham Bell. He had this idea of photophone instead of telephone. He encoded sound into a sunbeam. There was no laser yet. The sunbeam was propagated. At the remote end, you can convert the sunbeam into sound again through photoacoustics. Of course, now we're enabled by a set of modern concepts: laser, ultrasonics, tomography, and computer. We combine this set of concepts with photoacoustics to enable a novel technology, namely photoacoustic tomography. In photoacoustic tomography, we combine deep penetration of ultrasound with molecular sensitivity of optical contrast. We start with a very short laser pulse, nanosecond typically. We broaden the laser beam such that the energy per area is within the safety limit. So this is an extremely safe technology. We allow photons to walk in a random fashion. That's how you provide deep penetration. So we don't strive for ballistic photons. The ballistic photons disappear very quickly. When photons are absorbed, it doesn't matter if the photons are scattered or not. It'll generate very rapid heating. Every millidegree gives you a detectable signal already. Usually within the safety limit, you generate up to hundreds of milledegrees. So you get good signal to noise ratio to form a sharp image. Unlike standard optical imaging, we don't detect the returned photons. We detect the re-emitted acoustic waves. Because the acoustic scattering is orders of magnitude weaker than optical scattering, we can form a very sharp image as a result. Biological tissue is essentially transparent to ultrasound. However, we combine the acoustic resolution with optical contrast in a single modality. Without getting into the detailed math, I want to give you an analogy showing you how we can form an image. The principle is extremely straightforward. It's triangulation. Let's say we have thunderbolt. When you see lightning, you set your stopwatch to time 0. When you hear the thunder, you record the time delay, T1, multiply T1 by the speed of sound, V sub s. You get a radius for a spherical shell on which lightning took place. If you have 3 such measurements centered at different locations, you can triangulate and pinpoint the thunderbolt. Now, of course, in biological tissue, we don't have a single sound source. We have a distribution, 3D distribution of sound sources. Therefore, you need far more than 3 detectors to form a good image. In 2003, we reported the first functional, also the first in vivo, photoacoustic tomographic images in small animals with intact scalp and skull. By wiggling one side of the whiskers, the contralateral side was detected for functional change. You can see one side versus the other side. This paper excited our field for exponential growth. You can see it kicked off this growth right here. This paper has become one of the most cited papers in our field. Since 2010, the conference on photoacoustics has become the largest at a 20,000 attendee Photonics West. Since a few years ago, the field has been publishing over 1,000 peer-reviewed papers per year. Now, you do see this pandemic dip, I'm sure we'll pull out of very soon. Many companies have commercialized photoacoustic tomography. The technology has received FDA approval last year. Now, fast forward more than a decade, from 2003 to 2017, we finally evolved the technology from a single element transducer to 512 channels. We call this technology single impulse panoramic photoacoustic computed tomography. The system can be configured for truncal imaging. You start with the laser, route it to a conical lens. You form a hollow cone beam to efficiently deliver the laser energy to the small animal trunk. We use a full ring ultrasonic transducer to look at animal in every possible direction on a plane to form the best image. We call this full view image. We follow the Nyquist sampling criterion to minimize artifact. We use preamplifiers to maximize the signal to noise ratio before cable noise creeps in. We use 1-to-1 data acquisition, meaning 512 channels of data acquisition. Therefore, with a single laser shot, we can form a cross-sectional image. The data was acquired for a 2D image within tens of microseconds. So you have no motion artifact to worry about at all. We use a computer to reconstruct image and present image. Here's a close-up. You have this hollow cone beam. We have elevational focusing. This is very much like optical focusing, but using ultrasound, you can also focus the ultrasonic energy. That gives you elevational resolution in the Z direction. But the in-plane resolution comes from something called the inverse spherical redown transformation. So we have to reconstruct it to form an image. We can reconfigure a system for brain imaging. Here we can use a diffuser to form a solid as opposed to a hollow cone to efficiently deliver the energy to the brain. The rest of the system operation is identical to truncal imaging. Just give you one example where we can scan along the trunk of this small animal. The red line shows the current cross-section shown right here. Again, a single shot takes only tens of microseconds to acquire. And then we repeat our laser pulse. For this particular case, we repeat the laser pulse at 50 hertz. So you get 50 frames of images per second. Without injecting any contrast agents, we can see the structures in the body. Here, we're essentially detecting blood or hemoglobin as our contrast mechanism at this particular wavelength. You can tune our laser wavelength, of course, to image some other absorbers in the body. You can see pretty detailed structures, especially this close-up. You can see the kidney, the symmetry of the two sides of the kidney, as shown right here. Muscles, spinal cord, intestines, the heartbeat as well. We were excited about this initial success. We decided to scale up the system for human breast imaging. Now, of course, everything has to be a little bigger. This diameter is 22 centimeters in diameter, which accommodates most breasts, more than 95% of breasts. You can slightly deform the breast, which is painless, against the chest wall, just to minimize the depth requirement for light penetration. We started with 1064 nanometer laser. The rest of the systems are very much the same as the small animal system. You got 512 channels of data acquisition. We use preamplifiers as well to maximize signal to noise ratio. This system operates around this point. We plot the imaging depth versus the spatial resolution. Photoacoustic tomography is a technique that allows us to provide multi-scale imaging from organelles through tissue, through cells, tissues, all the way to human organs or whole body small animal organisms. It's a very unique capability because we're providing multi-scale imaging with consistent imaging contrast. We always detect light absorption. So at different length scales, we can correlate information acquired to offer a holistic picture of the biological problems we're studying. We believe this has a future in biomedicine. This dashed line roughly connects all the different implementations. No matter what depth you work at, you provide roughly 200 pixels in the depth direction. You provide high resolution imaging. So at this point, you provide centimeter scale penetration, maybe hundreds of microns resolution. This is one example from a healthy human volunteer. We use color to encode depth of the cross section. This is the center of the breast. With a single laser shot, you get a 2D cross-sectional image. The data was acquired within 150 microseconds. Within a single breath hold, 10 to 15 seconds, we acquire a 3D image by scanning away from the chest wall. The smallest vessel diameter we imaged was 1/4 of a millimeter. We provided 4 centimeter penetration. Again, this image was acquired without injecting any contrast agent. If you were to use MRI to form a breast image, oftentimes you have to inject gadolinium, which, as we know, is a heavy metal contrast agent. So these are different cross sections of the images. Let me give you one example of a breast cancer we detected. In this particular case, the breast was radiographically dense. Even in a 69-year-old patient, the breast was still radiographically dense. So it's extremely hard for X-ray to image. In fact, X-ray mammography missed the tumor. We used ultrasound to pick up the tumor, then we placed a metal target and took a second mammogram. The dot actually is the metal. By using photoacoustics, we can see this dense collection of blood vessels indicating the tumor location. We can analyze the vessel density. You can see the contrast here. Because we can image so fast, we can track the breast motion. By analyzing the motion, we can come up with a strain map. So this is essentially elastography. You get a second contrast for free. Of course, the more contrast you can provide, oftentimes the more accurate you can diagnose. Again, because of the high imaging rate, we can see dynamics of blood vessels. The postal motion can be analyzed. We can figure out as a result which vessels are arteries, which are veins. We use red to indicate arteries, blue to indicate veins. And you can see here the oscillations between the pixel 1 and 2 are vastly different. Because as we know, physiologically speaking, the veins don't really carry the pulsatile motion because they were filtered out by capillaries. We want to push our technology even further for 3D imaging, where we provide uniform spatial resolution and even brain imaging. So this is the system. In that effort, the project was funded for breast imaging, so it's not optimal for brain imaging. But we went ahead and used the system for brain imaging to get data anyway. So here we use 2 laser wavelengths that allows us to differentiate the 2 colors of hemoglobin, namely oxy and deoxy hemoglobin molecules. These are important because we can quantify oxygen saturation as a result. The system can operate in two modes, the anatomical mode, which takes 10 seconds to acquire image, where the functional mode takes only 2 seconds. We need to make it fast enough so we can track brain function. So the rest of the system is quite similar to the previous Bren system. The exception is that we have a bowl-shaped structure instead of a ring-shaped structure, and we have 1024 elements instead of 512 elements. We rotate the arcs. There are 4 arcs distributing the 1024 ultrasound transducers, and we rotate it to form a hemispherical detection geometry. Then we run 3D image reconstruction to form an image. This is an example breast image. Again, the key advancement is that now we can provide isotropic spatial resolution, so you can see blood vessels in detail in all 3 dimensions. Then we moved on to image human brains. We started with hemicraniectomy patients. So their skull was temporarily removed to release their brain pressure. And we borrowed those patients for our initial test. Notice here, when the finger started tapping, you can see brain activation right here. So this is the first functional photoacoustic tomography of human brains. In the next slide, I'll let our collaborator narrate how we compare with the strongest B field, magnetic field, namely 7 Tesla functional MRI.
Here we present results from subject 1, 7T MRI on the left, functional pact on the right. Individual functions are flashed in, starting with finger tapping, lip puckering, tongue tapping, passive listening, and silent word generation. Vessels for co-registration are then labeled 1 through 4, vessel 1 and 2 being superficial temporal artery, 3 and 4 being cortical arteries. The images are overlaid, and then individual functions are flashed in, starting with finger tapping, lip puckering, tongue tapping, passive listening, and silent word generation. The vessels are again pointed out individually, vessel 1 and 2 being branches of the superficial temporal artery, 3 and 4 being cortical vessels. The lateral perturbations allow for visual spatial appreciation of the co-registration. The images are then separated, and native T1 masks are faded in. Again, 7T MRI on the left, functional pact on the right. Individual functions are then flashed in, starting with finger tapping, axial, and coronal images through the area of interest are introduced below. Next, lip puckering, tongue tapping, passive listening, and silent word generation.
The big question is, why do we need photoacoustics? We already have functional MRI. In comparison to functional MRI, photoacoustics provides richer contrast, faster response, lower background, higher linearity, greater portability, open platform, lower acoustic noise, much lower cost, and no magnet. So there are a number of reasons why we need to provide new imaging modalities to complement this very well-established great modality, functional MRI. Now, of course, the big question is, we started with hemicraniectomy patients. Can we move forward? This is our next effort. We call this TransImplant functional photoacoustic tomography. This patient, after we put in this acrylic implant, we can still get functional images. This is very preliminary. We're still gathering more data to support it, but I want to share this excitement with you. Finger tapping showed responses here. Of course, the ultimate goal is to image normal human subjects. We call this transcranial functional photoacoustics. For the same craniotomy patient, hemicraniotomy patient, we image both sides. Again, you can see here the left side is hemicraniotomy without the skull bone. The right side is going through normal skull bone. We can still see functional response. So this is extremely exciting for us. We're increasing our patient population for more tests. As I mentioned earlier, photoacoustic imaging can be scaled for microscopic imaging as well. This photograph shows the first 3D photoacoustic microscope. This close-up shows the key components. We have a hollow cone beam that's refocused into the tissue. On the tissue surface, we have a donut beam. And the core of the beam was made dark intentionally to minimize surface interference. The acoustic detection is confocal with light delivery to maximize the signal to noise ratio. With a single laser shot, we get a 1D depth resolved image, very similar to a radar or ultrasound A-line, where you get a depth resolved image depending on the time of arrival of your signal. We can raster scan this system in a water tray, water-filled tank to get a 3D image. There's a membrane that allows us to couple light and sound. The object is placed below the membrane. Now we moved from here to this point where we provide much finer spatial resolution. It provides tens of microns spatial resolution, but you give up the penetration. We're still above or deeper than the optical diffusion limit. So this version provides roughly 3 millimeter penetration. This is an example image of our human palm. You can see blood vessels again without injecting contrast agents. If you only use X-ray to see blood vessels, you oftentimes have to inject iodine or another heavy metal type of contrast agent. So blood is a natural contrast agent for optics. You can look at the B-scan images that gives you the standard skin structures. Another application of our technology is to look at circulating tumor cells. Currently, there's no technique that allows us to image in vivo CTCs. Using photoacoustics, we started with a melanoma patient because their melanin is a natural contrast agent for us because melanoma cells tend to have enhanced melanin concentration. We took our device to the medical school. In this particular patient, you can see a bright spot swimming across a blood vessel. In the same patient, interestingly, you can see this doublet moving across. We capture them multiple times because we can provide enough imaging frame rate. It allows us to see them multiple times within the field of view. So by optimizing the optical wavelength, melanin becomes a much greater contrast than even blood. So this stands out with a great contrast. Now you can imagine all sorts of potential applications if you have a non-invasive tool like this, from screening to prognosis, diagnosis, and monitoring of therapy. And we can scale even further for better spatial resolution, but at the expense of penetration. So there's a little price to pay. With this version, we're talking about 1 millimeter penetration, but we're able to image single red blood cells, and we can watch in vivo how cells split at this juncture. It's not totally random, as you can tell. They tend to travel as a group. We can image humans. This is an image of our own finger cuticles. We can use pseudocolor to indicate oxygen saturation of hemoglobin. As oxygen is released, the color will change, as shown right here. This is one of the cells we singled out from this group of cells. So this provides a tool to study the fundamental physiology of oxygen release by a single cell. As we know that histology is the gold standard for disease diagnosis, but it requires tissue excision, requires pretty lengthy preparation before pathologists can examine the slices under a microscope. We want to do label-free photoacoustic histology. We don't have to label them because DNA and RNA in cell nuclei provide a natural contrast. By tuning the laser wavelength, we can target DNA and RNA absorption. That gives us the contrast very much similar to HE staining. So this is a photoacoustic microscopic image without staining. Our pathologists can diagnose out of our images already. They can differentiate normal from this human breast tumor. We validated our technology by sending this slice to HE staining. And you can see how closely we compare with the standard histology. We can also pseudo-color our images to make them look identical to HE staining. In fact, here, that's exactly what we did. Except we made one more advance. We want to image bone specimens. Now, bone pathology is extremely challenging because standard preparation takes up to 7 days. Because bones are hard to cut. You got to decalcify before you can cut. Using our technology, we don't decalcify. We don't label, we don't stain. So we can just reimage the raw sample, raw specimen directly. We use artificial intelligence to pseudo-color. You can see how closely they match. Again, we ask multiple pathologists to make diagnoses out of our images. They can make such diagnoses as if they're viewing standard HE stained images. We can even advance the resolution further to the organelle level. This is one example where we use very highly focused optical objectives to form a very sharp image. The standard optical diffraction limit resolution is 234 nanometers, where we're looking at a mitochondria. Now, using nonlinear photoacoustics, we can achieve a resolution of 90 nanometers. And this is actually super resolution. So sometimes we call this nanoscopy. And you can see a comparison right here. This is a EM micrograph. So I just showed you the scalability of photoacoustics over a vast length scale range. Now, after working on the length scale for decades, then we reminded ourselves, let's not forget the fourth dimension, if we call X, Y, Z, T, the four-dimensional space we live in. At the time, the fastest imaging technology was not fast enough to capture light motion. As we know, if Einstein is still correct, light travels at the terminal speed, at the final speed. Nothing can propagate faster than light. So we developed a technology called compressed ultrafast photography. The first version achieved 100 billion frames per second. With a single shot, you form an image. So this frame rate enables us to look at light propagation in real time. This is a single light pulse, so you don't have to repeat the light pulse to form a movie. The light pulse propagates in space, gets reflected, refracted, and you can watch two light pulses having a race. We can look at fluorescence decay in real time. Again, in real time with a single event. We change our camera to two colors. One color for the excitation pulse, one for the emission. And if you look at the green pulse, that was detected using the excitation channel or the green channel, and the red channel is for fluorescence emission. The fluorescence decays within nanosecond scale, but because we have picosecond type of resolution, and you can see that decay seems to be forever. You're watching the slowest slow motion movies because all the videos have been slowed down by 10 billion times. Now, if you're interested in FLIM, fluorescence lifetime imaging microscopy, you realize that this is essentially a single shot version of fluorescence lifetime imaging. And we can image even multiple decay constants. So how does this technology work? Let's start from the object with intensity I versus X, Y, and time T. So that's the movie you want to rebuild. We send the light to this digital micromirror device. that encodes a pseudo-random pattern. We call that C versus XY. These are ones and zeros that encode your light distribution. The encoded light is routed back and reflected toward this street camera. Unlike the standard operation of a street camera, we open up the slit as widely as possible because the standard operation uses a very narrow slit. They reserve the vertical dimension for temporal resolution. Because we want to capture the Y information as well as the X information, we open up the slit. So we let the Y information come in. We first convert photons into electrons through the photoelectric effect. Then when the electrons go toward the back end, we apply a voltage ramp. We call this shearing. As electrons propagate, depending on the time of arrival through this pair of electrodes, it'll experience different levels of shearing, the vertical shearing. So they'll be pulled to different directions. And that's a very smart way invented by the street camera folks to freeze time, essentially. So you no longer are under stress to image really fast because the temporal information is spatially encoded. Later on, we can just look at a vertical dimension and figure out the exact time of arrival. And that provides a very high frame rate. But again, using a standard street camera, you don't have the Y information because the vertical axis is exclusively used for temporal resolution. So in the end, we convert the electrons back into photons, then we use a CCD or CMOS camera to form an image. Everything I said can be combined into a single matrix equation. So the CCD will report energy matrix versus XY. So that's a function of the original coding operator, shearing from the street camera, and time integration within the exposure time of the CCD camera. All we have to do is to invert this matrix to form a movie. So you want to solve for I. But you quickly notice that I is three-dimensional. It has X, Y, and T as independent variables, where your detector is two-dimensional. You got X, Y only. So you got fewer equations than you have unknowns. As we know, we were taught right a long time ago, you cannot solve this equation. This is an underdetermined problem. However, the true number of unknowns is far less than the parent number of unknowns. And so there's this modern concept of math is called compressed sensing, where you can use that fact by solving the true number of unknowns. It's called sparsity. Using that concept, we can form a movie, as shown here. There are a number of applications. We started from basic physics. We know that when an object flies supersonically, you will form this so-called sonic boom or Mach cone. There's an optical counterpart, but it was never observed because there was no technique that allowed us to capture this. So we simulated this superluminal Mach cone. We have this medium here that we have a tunnel in the middle, which is not shown here. So light propagates faster in the tunnel than in the medium. That's superluminal, meaning faster than the speed of light in the medium. You can create an optical boom. This is a single shot observation of that optical boom. And this is the mechanism of Cherenkov radiation, which has some medical applications. As we know, phase contrast is valuable because sometimes, like a cell, it doesn't really give you amplitude or intensity contrast unless you stain. So in a native state, sometimes you have to depend on the phase as your contrast. And we incorporated phase contrast into our technique. This is demonstration. You can see this shock front propagating creates phase changes. This is another physical phenomenon. It's called chaos. Now, optical chaos runs at optical speed, and more importantly, it cannot be repeated. That's why it's called chaos. So using a standard pump probe, which requires repetition of the event, you cannot study chaos. So using our technique, we can study this quite well. This mushroom, half mushroom based cavity is actually a chaotic cavity. And if you watch this light pulse, this is a single light pulse we're watching in real time. We have two takes here. If we overlay the two movies, you can see at the beginning, the two pulses are overlaid almost exactly. But if you watch longer, the two pulses will split. And we did not change anything that because the cavity is chaotic, the pulses will split. Now, to study the transition between non-chaos and the chaos, we can build a light speed switch right here. And so this box is actually a stable cavity, but once you open up this wall, it's a gate, it becomes a chaotic cavity. So you can study the transition quite well using a real-time device. So this compressed ultrafast technology is a family of different implementations, and the speed has increased even further. So this is a plot of sequence depth versus the imaging speed. Sequence depth means number of frames you can capture per movie. Cup is in this place. This is the preferred corner of this plot. So you want to have high imaging speed and also long imaging sequence. The most recent reported speed from a lab is 70 trillion frames per second. And this is not even the ultimate limit, I don't believe. So we're pushing that limit even further. All right, let me quickly thank Caltech for building me a dream lab. This is one of the floors of our lab. We have omniscale imaging from nanoscopic imaging all the way to patient imaging. We do have openings for students and postdocs. Credit goes to lab members. We're funded by mainly NIH, but partly by NSF. We have a couple of books here if you're interested in more details, and our website has some more information as well. Thank you so much.
Thank you very much. Thanks very much, Dr. Wang. Fascinating material. We have several questions. If it's okay with you, I'll go ahead and read them out to you. Okay.
Please. Yeah.
Anthony Lamontier, one of the faculty here, asked, what are the limits of the XY field size for PACT? Can it be varied by the equivalent of different magnification lenses for optical microscopy?
It's a great question. So this is similar to the lens scale. If I can reshare that slide, it might be helpful. So there's a scalability here. Share. And somehow get in this mode. Share. Share screen. All right. No, it's not giving me this. Oh, it's on the one slide. Give me a second. Go to the right slide. We won't share. Screen. Yeah, this slide shows the imaging depth versus spatial resolution. So depth is the hardest place because your light attenuates, ultrasound attenuates. XY is actually a matter of effort. So the lateral dimension you can scan, for microscopy, for example, you can just scan forever. You can get a very large field of view. Now, of course, depending on the configuration for breast, you have this cylindrical geometry, and you got 22 centimeters in diameter. So that can limit you as well. So depending on applications, we're actually interested in building a whole body version, but here we switch laser light into microwave. So we can use a safe dose of microwave, because microwave, very similar to MRI, as we know. MRI uses RF. And so we can use the right RF frequency to provide whole body level penetration. There you're talking about really the waist size of the normal person as your field of view. I hope that answers the question.
Yeah,
so it's highly scalable. That's the beauty of this technique.
Yeah, thank you, Dr. Wang. I know Dr. Lamontier is very interested in having wide fields to do this kind of thing in the tissue they look at. So thank you. All right, next question is from Kenneth Young. He said, when you're using the acoustic detection technology what exactly is being detected? Is it always exogenous contrast agents, or does endogenous chromophores play a role?
So here we actually detect mostly endogenous, like hemoglobin. So the blood vessels you see are because of the hemoglobin absorption. When light gets into the body, the molecules absorb light. Depending on the wavelengths you tuned, so normally hemoglobin is a strong absorber, but for that circulating tumor cell detection, we actually image melanin. So we tune the laser wavelengths. At that wavelength, melanin absorbs much stronger than hemoglobin even. And so melanin signal stands out. Now you can tune the laser wavelength to image all sorts of other molecules. I don't have time to show. Just to name a few examples, you can image myoglobin, right? You can image lipids, water. Of course, the histology images I showed are DNA and RNA absorption. Now, if you're allowed to introduce exogenous contrast agents, we've done, of course, using other techniques, using other contrast agents as well. You can image nanoparticles, you can image proteins, like all those fluorescent molecules are also absorbers, because the fluorescent quantum yield is almost never 100%, right? So usually you get 10%, you're pretty happy. In fact, many of our collaborators, they just failed when they try to develop a good fluorophore. They say, oh, my fluorescent quantum yield is terrible. Let me talk to Lihong. So, because we can image essentially any molecules, whether it's fluorescent or not. If it has fluorescence, one minus the fluorescent quantum yield actually gives us the signal. And oftentimes we get a bigger portion for photoacoustic imaging. Now, of course, even when the molecule is non-fluorescent, we can still see them. As we know, in our body, most molecules are non-fluorescent. We got a few that are fluorescent. So you can have reporter genes imaged using photoacoustics, organic dyes, nanoparticles, you name it. Anything optical absorbing at any of the wavelengths we have can be potentially used.
Great. Okay, thank you. Next question is, when you use the PAT setup that was originally used to scan and image breast cancer to map functional activity in cerebral tissue during tasks like finger tapping, did you change or modify the ultrasound transducer or the amplitude of the wave due to skull bone physical characteristics, the laminar structure of the cortex, attenuation of radiation force, or distortion of local point length caused by the skull on specific target tissues?
That's a very good question again. So we started with the easy case. So we started with hemicraniectomy. Originally, we want to start with neonates where the fontanelles are open, but neonatal patients are very hard to recruit, as you might be aware of. And so our collaborator said, hey, we have this spatial patient population. These are hemicraniectomy patients, and they're available, because usually they remove the skull bone. They usually have some head trauma, so they build up their brain pressure. They want to release the brain pressure. The surgeon just temporarily remove the skull bone, and usually, a few months later they'll put it back. They used to use the original skull bone, but they actually found out using implant is actually better. So nowadays they use implants, and so that gives us opportunity because we can image without the skull bone and with the implant, as we demonstrated recently, and even on the normal side through the skull bone. The skull bone actually presents a challenge because it attenuates both light and sound by roughly order of magnitude. And so we have to improve our system. Relative to breast imaging, we have to work like order of magnitude harder in order to get equivalent type of image. And as shown by the preliminary data, we can do that. And so there are many ways to improve. The system we use to demonstrate the initial images of brain function is optimized for breast imaging. So for brain imaging, it's working at the wrong frequency. For brain imaging, it turned out 1 megahertz is the optimal choice because the skull bone attenuates very severely at high frequency. For breast imaging, we picked 2.25 megahertz. So at that frequency, a lot of the signal will be attenuated too much. And so luckily the transducers have broadband. So even at 1 megahertz, we still get some signal going through the skull bone. We are building the next version, and the hope is that the signal to noise ratio is going to be far better. So it's all about signal to noise ratio. Now, the other challenge is that the skull bone is like a bad lens. When we look through a bad lens, of course, all the images will be distorted. So there's acoustic aberration involved in adult transcranial imaging. And so there are ways to de-aberrate. But even without de-aberration, we can still form a sharper image. This is different from ultrasound imaging. With ultrasound imaging, your ultrasound pulse goes in, you suffer from one distortion, and the ultrasound signal has to come out. You suffer from another distortion. So it's a round-trip distortion, round-trip attenuation, and makes transcranial ultrasound imaging very, very challenging. And luckily, our ultrasound signal, which is the image formation part of the signal, only goes through one-way attenuation or one-way distortion. And so we can actually find ways to correct for those distortions. And we have less distortion to begin with. And you may have noticed that unlike ultrasound imaging, we don't have strong spectral artifacts. And so we have better images to start with, and then we can hopefully correct and even form sharper images. And physiologically speaking, for functional imaging, oftentimes they tend to be a blob anyway, because within seconds of response time, and the hemoglobin or blood flow contrast tends to propagate and they diffuse. And so you don't necessarily need a super high spatial resolution to see the functional changes to begin with. And so that's why we're very optimistic this may have a future.
Great, thank you. We have several questions along the same vein. I'm going to pick one of them out here from Dr. Zhisheng. He said one of the major problems in brain cancer is the lack of early diagnosis, given the high cost and potential side effects using MRI and routine brain scanning. So he asked the question, will PCAT be able to overcome these difficulties and be used potentially as an early diagnostic tool for brain cancers?
Again, a great question. So using light, we have a limited penetration. In breast, we can provide maybe one way of 4 centimeters. And with breast, of course, you can illuminate from both sides. So the total thickness could be doubled, up to 8 or so centimeters. With the brain, brain is more absorbing, and you got the hair in the way as well. So far we demonstrated about 2 centimeter penetration. So using light alone, you may not be able to see the whole brain, but we are building this RF/microwave version. And there's hope that this can potentially provide deep penetration of the entire brain. So, let's just wait and see if that can work.
Okay, exciting. One of our cardiovascular researchers, Dr. Steve Polsing, asked, can you image the double slit experiment with a photon and resolve whether the wave function passes through both slits, or is it always resolved through one of the slits because it's measured?
Well, thank you for that interesting question. You must have a physics background. We actually, this camera, as soon as we published this paper, right? So we thought about this direction. We got many inquiries about similar problems because the quantum nature, if you got a single photon, what happens there? You have 2 slits. If the photon is only allowed to go through one slit, why do we see at a time? Why do we see interference pattern, right? So, Dirac says the photon interferes with itself, so we have to respect that because there's no better answer. But people are looking for better answers. What's going on there? So you can see this is still an active research topic. People are still questioning the fundamental physics. Why is there interference if it's a real particle, it's a single particle, despite the duality, so that gives us justification. We can use either picture at will, whenever we need, whenever we can't explain one thing, let's just switch to the other one. But the classical world doesn't work that way. So how do we reconcile this difference? We have an active research area. There's this Bell's test you might have heard of. So that's a very interesting tool, under local hidden variable theory, and Bell derived this inequality. It turned out that photons can violate that, through entangled photons. So the jury is still out. I personally believe there's some very deep physics to be uncovered one day. So we're studying this. Now we have this tool, and our hope is that many more groups will join force and push this research direction as well. This compressed ultrafast camera is the only technique that provides both X, Y, and time t at light speed. So we can detect events, what's going on there. The theory to explain all the quantum phenomena is there's some communication, even when the two detectors are miles apart, somehow there has to be superluminal communication in order to reconcile, to figure out what happened to the detected signals in terms of their statistics. And so it's extremely a tricky physical phenomenon. I'm glad you raised that question. So double slit and all those other experiments, they all share the same type of mystery.
All right, thank you. And somewhat related, Anthony Lamonte asked again, he says, how can nanoscale imaging be used to identify and image single distinct molecules of choice?
So, all right, single molecule, you have to have enough signal to noise ratio, right? So if you have a single molecule, you only give me one photon, then it could be very challenging. So if you have a single molecule, you fire a stream of photons, you collect enough photon counts, there's a Shah noise, right? So if you collect 10 photons, your Shah noise is square root of 10. So you may have a good chance because your SNR is roughly 3 to 1, right? And so that allows you to see that molecule quite reliably. But as long as you collect enough photons from a single molecule, then you can detect it.
Okay. Thank you. And
Sure.
I think, based on time, we'll just do one more question. This one is, actually, I don't know who it's from. It says anonymous attendee, but anyway, could photoacoustic tomography be used in real time to monitor medical procedures, for example?
Yeah, we can actually make it real time. So the 50 hertz, for example, right? We can actually stream. So real time 2D imaging is possible. 3D imaging, sometimes you still have to acquire data. So depending on the number of transducers you have. So in theory, even 3D imaging can be made real time. So it is possible, because the data acquisition is very, very fast. It's limited by the acoustic transit time across your field of view. For some animals, it's tens of microseconds. For human breast, it's hundreds, like 150 or so microseconds, right? So even for a whole body trunk, you're talking about still hundreds of microseconds. So less than 1 millisecond, you acquire all the data. And the bottleneck is how quickly you can reconstruct the image. But for 2D, we can reconstruct very fast. For 3D, we have to resort to GPUs. So we can parallelize our image reconstruction and provide real-time imaging as well.
Okay, thanks. And actually, since we have one more minute, I can squeeze one more in. One of our graduate students, Gates Palisari, asks, she says, it seems like functional acoustics will potentially one day replace functional MRI. And also, do you think photoacoustics will replace 1 and 2 photon imaging in the very near future?
So if you look at radiology, all those modalities coexist. In biological imaging, it's the same. You got confocal 2 photon. You have flame imaging. So I believe nobody is going to replace any technologies, but they're all complementary. I listed a number of advantages that photoacoustics can provide over functional MRI, but functional MRI has also advantages. For example, whole body penetration, we're not there yet. So when the object is too deep, we don't have the right penetration. And so, the hope is that there is a place for every technology.
Okay. Well, look, Dr. Wang, thank you very much. Fascinating material.
Thank you. Thank you all.
Thank you. I will get a chance to talk to you a little bit later, Dr. Wang. I think you have some other meetings coming up. So.
Great. Thank you again. Bye.
Thank you.